Dr. Brenda Andrews and Dr. Charles Boone, both at the University of Toronto, have been collaborating for several years on a number of projects involving large-scale genetic network analysis. In their latest work, published in the January 11th issue of the Journal of Cell Biology, the group combined the automated yeast genetic method they co-developed with automated image acquisition and analysis. This allowed them to analyze their arrays using a cell biological readout to look at genetic interactions in budding yeast.
Genetic network analysis is this system we call synthetic genetic array analysis, or SGA analysis. And with that method, you can create recombinant yeast cells by crossing any marked mutation into an array of mutants or strains that are carrying other marked genetic elements.
In their paper, the group used this automated process to introduce fluorescently labeled tubulin into their yeast deletion collection. This allowed them to use spindle defects as their cell biological readout.
We have 15,000 different mutant combinations in the paper, the JCB paper. We looked at every deletion mutant, single mutant, for defects in the mitotic spindle, and then we also looked in 2 sensitized mutant backgrounds for defects in the mitotic spindle.
Each gene was therefore looked at as a single mutant and as a double mutant in either the BNI1 or the BIM1 deleted background. These spindle defects were quantified using high-throughput microscopy to capture and analyze the image.
And then when you image a field of cells, you can distinguish single cells using a computer-based analysis system. And once you can distinguish single cells, then you can start to measure various parameters like cell shape and size and budding index and things like that.
The group then focused on 3 parameters to identify genes that regulate spindle morphogenesis: the ratio of spindle length to cell length, the orientation of the spindle, and the distance between the spindle's center and the bud neck. Because these data are quantitative, it allowed them to use statistical methods in their analysis.
Every single image that we take, there's a couple hundred objects in there, so you're getting a couple of hundred data points for every single image that you take. So getting decent statistics is fairly straightforward.
By setting a p-value cutoff of 0.1, the group identified almost 2,000 mutants with at least one of the 3 spindle defects amongst the 3 screens. The group then whittled this number down to 182 mutants for further analysis. Amongst these were mutants that were found to exhibit hyperextended fishhook spindles, but were previously unlinked to spindle disassembly. Yeast strains defective for MCM21, HNT3, EMI1, and EMI2 exhibited increased number of cells positive for fishhook spindles in either the single mutant or BNI1 deletion double mutant. Time-lapse microscopy also revealed that these strains had delayed spindle disassembly as evidenced by the presence of fishhook spindles, indicated here by yellow arrows. This phenotype points to a possible link to one component of the chromosomal passenger complex called IPL1, the aurora B kinase, which is also known to result in hyperextended fishhook spindles. Further analysis revealed a previously undiscovered means for regulating the localization of IPL1P and therefore the chromosomal passenger complex during mitosis.
It looks like this chromosome passenger complex moves from the kinetis core to the spindle midzone, and that it seems to do so in a simulation-dependent way. And so we discovered that simulation of a kinetochore protein called MCM21 is critical for the movement of the chromosome passenger complex to the spindle midzone, such that it could then mediate spindle disassembly and, in a normal manner.
In addition to being regulated by simulation of MCM21P, further analysis also pointed to a genetic interaction between components of the kinetochore with components of the mitotic exit network and the 14 early anaphase release network. This proof of concept study reveals the utility of this integrated and quantitative methodology for uncovering previously unknown interactions.
So we were able to discover several new genes that had clear roles in mitotic spindle function, and then we also identified an equal number of genes by screening in sensitized backgrounds. So even in pretty well-trodden pathways, we could still discover many new things by doing these cell biological screens.
Dr. John Aitchison at the Institute for Systems Biology believes this is a really exciting next step.
So for a systems biology perspective, that's analogous to a microarray. A microarray is a continuous quantitative phenotype, in that case looking at transcriptional, or in many cases looking at transcription. And you can use lots of statistical methods and machine learning approaches to sort of cluster genes and identify genes that have related function based on those clustering algorithms. So basically you can do the same thing here. And you can identify combinations of mutants that lead to very similar phenotypes, and those that, in this case, the one they focused on was these fishhook type spindles. And so by that you can identify, you can directly see that each of these genes are likely involved in one specific aspect, and that is the formation of a normal spindle.
To find out more, please refer to the January 11th issue of the Journal of Cell Biology, where you will find the paper by Vizeacoumar et al., as well as a comment by Salim and Aitchison. You can also find out more by checking out the January 11th edition of BioBites.